Greetings, my name is GUAN, matrix number is 206974. In this video, I would like to demonstrate the technique of gram staining, including the preparation of bacterial smear. These are the materials and apparatus that we needed during this technique, and due to lack of material and apparatus, we will use other material and apparatus to carry out the technique for me to explain the principle. Before we stain the desired bacteria, we need to prepare the smear. This is because the smear will fix the bacteria onto the slide and prevent it loss during the staining procedure. A good smear need to prepare, or else it will give a poor result due to retention of dye if too thick, or lack of cell and waste more time and energy to find it if too thin. Without further ado, let's get started. A smear can prepare from solid or groove medium. If from groove medium, first flame the inoculating loop to sterilize it, let it cool down. Open the cap of the groove medium, flame the neck of the bottle, and get a loop full of the bacteria, and close the cap, flame it. Then place it onto a clean slide and spread it with circular movement until appear thin layer like a size of a 10-cent coin. While for the solid medium, first flame the inoculating loop to sterilize it. Then get 1 to 2 loopfuls of sterile water and place it onto clean slide. Then transfer small amount of bacterial inoculum from the medium onto the slide just now, and spread it into thin layer and like a size about a coin. Then allow the smears to air dry. At last, hold the slide at the one end and then quickly pass the smear over the flame of the Bunsen burner 2 to 3 times. Note that it is not direct heat, as it will distort the shape and the size of the bacteria. After the bacterial smear were prepared, then the slide are ready to be stained. First, the slide was flooded with crystal violet primary stain for 1 minute. And then wash off with tap water gently. After that, flooded with gram iodine as a mordant, also for 1 minute, and then wash off gently with tap water. After that, the smear will decolorize by using 95% of ethyl alcohol until it's run out clear, and then wash off with tap water gently. Lastly, the smear will counter stain with safranin for 45 seconds. And then wash off with tap water gently. Then the slide can be blotted dry by using debulous paper gently. And then the slide can be examined under oil immersion. This is an example of expected result for gram staining under oil immersion 1000 time magnification. In the image, the Staphylococcus aureus show gram positive, which is purple in color, due to its thick peptidoglycan cell wall, while for the Escherichia coli show gram negative in pink color, due to its thin layer of peptidoglycan and outer membrane with a lipopolysaccharide component. One thing that should be noted during the staining process is if alcohol rests in very long time, it may give poor result as it overly decolorize. For example, the gram positive will become
gram negative.